Fantastic.
Okay. So welcome back to part two of our ASO design workshop. Today we'll be focusing primarily on gene knockdown and allele-specific knockdown ASOs, but just wanted to recap from last time the different types of ASOs. Um, so the splice modulating ASOs, like we discussed before, it's a very similar strategy for exon skipping, exon inclusion, or splicing variants, where the ASO targets and redirects splicing. But gene knockdown ASOs work in a different way. They have a different structure where rather than just being modified RNA, they have an RNA DNA RNA structure with like a gap of DNA in the middle that is used to degrade the mRNA. So they work by binding to either the pre-mRNA or mRNA, depending on if you target an intronic or exonic sequence, and they recruit RNase H, which is an enzyme that cleaves and degrades the mRNA. So they can be used to knock down a gene, either both copies or one copy, depending on how you design them. So here's kind of an overview of our KCNT1 knockdown ASO project. As I think you guys know, we have an ASO called valeriocin that is currently in clinic that we've used to treat 2 patients with KCNT1 epilepsy. This is the process that we followed to get that ASO to the clinic. We designed thousands of sequences, screened over 200 of them in patient cell lines, and over 30 of them in rodent studies, 3 of them in a rat toxicology study to ultimately get 1 lead. So compared to our splice modulating projects, the GAPMER projects require a lot more high throughput ASO screening. It takes a lot more to find a sequence that is working well, depending on your gene. It seems like some genes, the sequence works better than others, and we can get away with screening less. And we also run into higher toxicity with these ASOs. So it's really important that we have a decent number that are working well in vitro, that we can bring into the in vivo studies, because it seems like maybe 1 in 5 or so will be tolerated in the acute tolerability studies. So GAPMER ASOs, like I said, can either be specific to the whole gene or to a specific mutation, to knock down in an allele selective manner. So if the disease is caused by dominant negative mutations, you would want to use allele specific knockdown. So if the wild type allele is creating a normal protein, but the mutant allele has a dominant negative mutation and is creating a mutant protein, you can design the ASOs specifically to just the mutant allele. So you're still creating normal protein, but degrading the mutant allele, leading to haploinsufficiency. So this is for diseases where we don't believe that haploinsufficiency leads to a severe disease phenotype. In cases like KCNT1, where we're doing a complete gene knockdown, that would be for cases with gain of function mutations. So when we're designing allele-specific knockdown ASOs, we use long-read sequencing to identify haplotypes in our patients, and this can help us discriminate between the wild type and mutant alleles. So in a group of patients that have a SNP at one site, you can see that they may have different haplotypes of different linked SNPs that are also on that allele. So we utilize these to expand our design space beyond just the disease-causing mutation. So the white box is the wild type allele here, and then below is the mutant allele. So if we were to just design the ASOs against the dominant negative mutation itself, we would be limited to only being able to target this one region of the gene. So the ASOs are usually around 20 base pairs, so you would have just that region from like 20 base pairs to the left and 20 base pairs to the right. But once we identify the haplotype sequence, we can then expand our ASO design space across the allele to other benign polymorphisms that the patient has in phase with the dominant negative mutation. So these would just be benign SNPs, and some of them may be common across the patient population. And once we know that they are on the same allele and are heterozygous, not also on the wild type allele, we can design ASOs against those as well, which helps us to expand our search. We can also utilize the benign polymorphisms on the wild type allele as well by targeting the reference base at that site. So if there's a heterozygous SNP that phases away from the dominant negative mutation, if you were to target the reference sequence at that site with an ASO, it would also be specific to the mutant allele. So now we've like greatly expanded the spots in the gene that we can target. This is also a useful strategy if you're trying to identify a more common drug target to treat a larger population of patients. So for a lot of these ultra rare diseases, the disease-causing mutations themselves are also ultra rare, and there may only be one patient in your cohort that has a particular disease-causing mutation, but you may have many patients that have this disease. So if we were to design an ASO specific to each patient's disease-causing mutation, we would need 3 ASOs to treat 3 different patients. Whereas if we were to target the linked more common polymorphisms, which can be present in up to 50% of the population, we can now find ASOs that could be used to treat 2 or 3 out of 3 patients if we find phased SNPs that they all share.
Amazing. Okay. So I'm going to touch briefly on a few considerations for how we optimize these ASO drugs and how we think about toxicity and selectivity. These can be kind of tricky chemistries and tricky structures. So I just wanted to give some background here. But to start, this is something I believe we touched on last week. This is a tool that we've been using and running in our lab from this paper that was published in 2022. We just refer to it as the Hagedorn score because Hagedorn is the first author on this paper, but it's a calculation that helps to predict toxicity of a given ASO sequence. It's not a perfect prediction by any means, but it's something that can help sort of give a first pass screen if you have a large list of designs to determine if a sequence might be more or less toxic when it moves into an animal model. So this is the paper, it's linked here. I would definitely recommend giving it a read. It was a really interesting sort of model that they put forward. But this is the calculation itself. So it takes into account essentially just the ASO sequence. So the number of each type of nucleotide that's present in the design. And then it also considers the position of the last G in your sequence because Gs are pretty important to toxicity, we found. So you would kind of count up the number of each nucleotide and then count where the last G is from the 3' end of your sequence. And then this is the calculation to determine this sort of predictive toxicity score. These coefficients were found from this group. They trained a model using mouse tolerability data and sort of developed this framework. And it's available both in an existing piece of R code that they have put in their supplement, and also you can just kind of pop this into Excel and use this as an Excel formula. We have a version of it that we're happy to share. It's a little funky, but it works pretty well. And one thing I did want to note is that the model was trained on LNA ASOs. So that model is not necessarily as applicable to all ASO types. We have to remember that all ASOs aren't created equal, and so training a model on one specific type doesn't necessarily mean that it's perfectly applicable to all other ASO types, like a full MOE ASO or an ASO that doesn't have the LNA chemistry. So just something I wanted to note, something that we think about in our lab as, okay, like how are we using this score and are we using it as appropriately as we can. But it is a very simple calculation and one that you could run really quickly on all of your designs if you wanted. And we find it's just a nice way to kind of filter out some possibly toxic sequences. And just to touch briefly on sort of the cutoff for this score. So this is a figure from this Hagedorn paper, which is comparing the tolerability on the x-axis in mice, and then on the y-axis, it's showing the calculated score from this equation. So the green space is representing an ASO that is well tolerated in mice, and then the orange is sort of like a toxic ASO that was not well tolerated. And you can see that there's a pretty strong correlation between their predicted score and how well an ASO performed in mice. They showed that with a cutoff of 70, they had like 90% confidence that that ASO, if if an ASO scored below 70, that it's predicted to be toxic. If an ASO scored above 70, then it's predicted to be hopefully safe. Unfortunately, none of the ASOs that we've designed have kind of reached this cutoff of 70. They tend to be a bit lower. So we have kind of thought about a score of 30 as a general benchmark based on some of the previous mouse studies that we've done and some of the results that we've gotten back. If an ASO is kind of floating around 30, we think that it has a decent chance of being tolerated. If it's significantly lower than 30, we might be a bit more worried about um, its its toxicity. So this is by no means a strict benchmark. I think we definitely have seen sequences that have fallen on either side of that and kind of been against the pattern, but just something that we're kind of using at the moment for trying to filter out some of these designs. But with more designs and more mouse studies and things like that, it's possible that with time we would get sequences that would fall closer to the 70 cutoff, which would be great. But so far, this is kind of the benchmark that we're using. All right. So the last little section I wanted to touch on is just some of the options that we have to make an ASO more selective or less toxic. And this kind of gets more into ASO chemistry and certain modifications to ASO design, which can be a little bit technical. And we have not explored all of these options yet in the lab. A lot of this is like new to the literature, but just wanted to include it here as options as ASO design projects move forward and as your pipeline maybe is growing. Okay, so first I just put a summary slide of the different types of modifications that we most commonly think of when we're looking into ASOs. These are probably things that you've maybe heard of when, I don't know, if reading about ASOs or ASO papers. So you can have fully unmodified DNA and RNA, and then there are additional backbone modifications or 2' sugar ring modifications that you can add on to that. So almost all of our current ASO designs use this PS phosphorothioate backbone, which is this is a form of DNA and is especially useful with RNA-seq activity. So like our Gapmer ASOs are using this backbone. There's also like morpholino ASOs, which you might have heard of. There are definitely some examples in the past in the literature that have been successful with morpholinos. And then on top of that, there's also these 2' modifications. So the locked nucleic acid, this is the LNA structure that I was talking about, the Hagedorn paper using. And then there's like 2' OME, 2' MOE, which are confusing because they sound very similar. They're just two different types of sugar ring modifications. And we use these MOE ASOs pretty frequently during our screening. But both methods are possibly useful. We found MOE are the best when moving a design to the clinic, but LNAs also have their advantages. And when looking at allele selectivity, you could consider adding LNA chemistry and things like that. All right. So in addition, there's sort of a newer paper that came out last year by Siddhar Agarwal. I'm not sure if any of you have seen this or have had the chance to read it. I did put the link here in the slides. And this highlights sort of like additional types of ASO chemistries and sort of where the next generation of RNA therapeutics is going. And I thought this figure was kind of helpful in just giving a brief overview of some of the types of ASOs and sort of the structures that that we've seen in the past. So you can have a full PO backbone DNA molecule. You can have a full PS backbone DNA molecule. You can have a molecule that's entirely 2' RNA based, which these tend to be what most of our like splice switching molecules, PS molecules are. You can have a gapmer, which this is what Aubrey was discussing at the beginning. So having a stretch of RNA with DNA gap in the middle, and then another stretch of RNA on the other end. We refer to these RNA chunks as the wings of the design, and then the gap in the middle. There's a splitmer, which is essentially just like half RNA, half DNA. This is definitely a slightly newer design that we haven't really used extensively yet, but is kind of up and coming. And then also this like cyclic structured, having a splitmer also attached to this other PO kind of tag that like loops and like rebinds to itself on the other side. So this cyclic structure is not something that we have worked with yet, but again, is like sort of a new idea that has been sort of posed in the literature and is something that I think is interesting and could be really helpful for reducing toxicity and also making designs more selective to a specific mutation.
Like Emma said earlier, like in the Hagedorn score prediction, that 3' end of the
Mhm.
ASO sequence is important, we believe, in predicting ASO toxicity. So this cyclic structure masks the 3' end of the ASO when it's taken up into the cells. So that's part of the justification, I think, for
Totally.
how they came up with this, and how it improves toxicity.
Yeah, absolutely. Amazing. All right, and then I believe this is the last slide, just on sort of ASO structure. And I know this figure is a little bit busy, so no need to fully understand every piece of it, but we like to think about, essentially the takeaway from this slide is that we like to think about how long the wings are and how long the gap is in a gapmer ASO. So this paper showed that a gap of 7 nucleotides that has the mutation kind of in this underlined position here and like the third position of the gap um, was maximized the allele specific knockdown effect because it blocks these kind of minor RNA sites and make sure that the cleavage is happening right next to the SNP, which was shown to be effective. So this is just kind of another design structure that we think can be really useful. This is a shorter compound because it's only, well, I guess it's like 15. Usually ours tend to be closer to 20. So this is a bit shorter. You would have to really make sure you're checking off targets and making sure that this isn't binding to another area that's really important, but is another sort of design that we think could be useful. And then a couple other notes at the bottom. It's been shown that ASOs with higher number of MOE nucleotides are thought to be less toxic. So if you can kind of maximize those MOE bases, that could be really helpful. But it also has a higher melting temperature than DNA. So kind of have to balance how you kind of count your wings versus your gaps and what RNA, DNA are present. You can also kind of stagger. So put another DNA molecule in the middle of a wing. That's been shown to possibly be helpful. You can add a second mismatch into a sequence, which has also been shown to be helpful for toxicity. So kind of a few different methods that we're still playing with and definitely don't have all the answers for, but some designs that have been shown to be interesting and helpful. Right. Okay, what time is it? Okay, I think I'm going to take a brief pause for any questions. And then there is this small design tool example that was written by someone in our lab that I can show you how it works. But honestly, um, Taehoon's tool in Python script that you showed before is basically the same thing and I think is as effective. So I'm not even as worried about showing that. But yeah, let's take a brief question break and we can discuss anything about the slides or anything about gapmers that you guys have questions about. And then I can show you the video if you want, but it's pretty much the same thing. It's just like running ASO design without having to do it manually. So pretty simple app.
I'm sorry, I do not have any questions, but I'm
That's
just
okay.
listening, following. Yeah,
Yeah, it
yeah,
was a very fast overview of a pretty complicated
Totally.
process. So if you have questions that come up once you try it, feel
Yeah.
free to reach out to us.
Yeah. We can definitely share our slides and can poke through and take a look at some of the resources too. Okay.
Well, I guess
Cool.
we can show you the video.
Yeah.
This is a tool that one of our research assistants designed. It's currently hosted on a server that you have to like activate before you run it. So it's not like a website that we can easily share, but
Yeah.
Taehoon, if you are interested in designing tools like this and want to work with us on making this more widely available or a version that you want to design, we're happy to collaborate.
Absolutely. Okay. I think I am now fully sharing my audio too. Okay. So yes, this is a tool that's run from the command line. It's mostly written in Python. And he's just going to run 20 mers for a region, a specific gene. Here, wait, let me pause it because this is a lot. All right, so the tool runs just tiling across a region. So it's doing every single base of a 20 nucleotide sequence. And then it has columns built in for GC content, melting temperature, Hagedorn score, off targets, all the things that we've kind of been talking about as necessary. And then he'll go through and he's going to filter those columns based on some of the criteria that we've discussed. So that's what the next steps will be to kind of it's essentially tiling across a whole region and then sort of filtering down to look for sequences that are useful. It also has the like splice enhancers, splice silencers built in. Honestly, not entirely sure which source he was using to call those. Probably ESC Finder if I had to guess. And then he's going to run off-target analysis for those 7 designs that passed filtering, which takes a little bit of time, but
I think it's just using the GG genome API a little bit for it to.
yeah. So he's going to export this to an Excel file. But just to note, there's like one call with 0 mismatches for all of these designs, which makes sense because it's calling the target. It's just calling the sequence that we expect. And then it shows 1 mismatch and 2 mismatches as well, and how many hits there are. I think he might show us the file. So it's nice that this exports to an Excel file, and then you can kind of use that for filtering or for just taking a look. So kind of an example output of what this type of tool might look like. But again, very similar to what you've already done and kind of same process. And then I think what he'll do next is there's another feature here for looking at an allele selective approach. So thinking about targeting a mutant sequence specifically. And so I think he'll run that as well, which won't look that different really, but is able to focus on just a target allele.
So for this, the 2 input files that he just selected were from the patient's long read whole genome sequencing data. We had a haplotype FASTA file for the mutant allele and one for the wild type allele. So now it will tile the entire sequence for both of those and then filter out any ASOs that overlap that would match up to both alleles, leaving just the ones that are specific to the mutant allele.
ran off targets again. That's it. Yeah, just did that. Cool. Okay, let me get out of this. And that was our last slide anyway. So, fantastic. Okay. So yeah, that's everything that we had to share with you all today. And I think hopefully this was a decent overview of some of the ASO design sort of types and what we tend to think about when we're looking for targets. But yeah, always here to answer questions and happy to discuss it more at any point, or if you guys want to give it a try and take a look at a Gapmer case, always, yeah, happy to discuss.